All right. Good evening, everyone. I think it's time to get started. It's my real pleasure to welcome you all to our distinguished seminar series on frontiers in biotechnology. This seminar series is sponsored by Invitrogen, and we are very grateful for that sponsorship, and we want to thank Invitrogen for hosting this seminar series, and this will be the inaugural lecture. So thank you, Invitrogen. Now, we are really privileged tonight to have Bob Langer with us. He's one of the icons in biotechnology in this country. And as you know, we have a national academy here in this country, and it is one of the biggest honors we can achieve as engineers to be elected to the National Academy of Engineering. Some of us get elected to the National Academy of Sciences. And there are only 6 people in the country that are elected to all 3 branches of our National Academy, to the National Academy of Engineering, the National Academy of Sciences, and the Institute of Medicine. Okay? And we have 3 of these 6 people right here tonight. This is a very special occasion. So I would like to ask... I would like to ask Bert Fung, Shu Chen, and Bob Langer please to stand up and give them another round of applause. When we announced this lecture by Bob Langer, the response was absolutely tremendous. We had over 100 RSVPs in the first hour. Okay. And so that just shows the kind of person Bob Langer is, and you will hear much more about him when he gets officially introduced afterwards. But certainly we are very pleased to have him here tonight, and so in terms of logistics, I will in a very short time introduce Claude Benjamin, who has been the moving force on the Invitrogen side. And then he will introduce Professor Shu Chen, who is the founder of our bioengineering department here at UC San Diego. And then Bob Langer will give us the honors, and then David Brenner will close by taking care of the question and answer session. David is our Dean of the School of Medicine, our Vice Chancellor for the Health Sciences. So this will be the evening, and then afterwards we have a coffee and cake reception out there. So I hope all of you will stay for a little bit of time to talk with especially our special guest, Bob Langer, and with the rest of us. So with that, I want to thank one more time Invitrogen, especially Claude Benjamin, who made it all happen, and then also Bruce Branchard, who was instrumental in that, and then from UCSD side, Shu Chen and Sadiq Esner. Those were the 2 who really made this special lecture tonight happen, so thank you to all of you. And so with that, I would like to ask Claude Benjamin to come up here and say a few words on Invitrogen's behalf. So Claude.
Thank you, Frieder. I'm very pleased and very excited to also welcome to the inaugural event of the Invitrogen UCSD Frontier in Biotechnology Distinguished Seminars. Tonight is the result of a shared vision between Invitrogen R&D and the UCSD Jacobs School of Engineering. This program began as an idea that Dean Frieder Seible and I had to develop an innovative educational program at the academic-industrial interface, particularly around how cutting-edge engineering technologies can be applied to challenges in the biotechnology and life science industries. So following through on this goal, you can imagine a ton of people worked to make this happen, and they're all here tonight, and without them, we would not be here. So on both sides, there were a lot of people who just the first thing is find a date was already a major challenge. But it's amazing that we were able to get this working. And it's a reality today. It's very exciting. So why this? What's the vision here? The vision that we shared, that Frieder and I shared, and with some of my colleagues at Invitrogen and some of the professors at UCSD, there were around 4 objectives. The first one is to strengthen the synergistic alignment between 2 leading institutions and their respective missions. Invitrogen is one of the premier biotechnology companies in the world, and a company that continues to define what biotechnology is by the area we work in and the products we sell. We are repeatedly ranked #1 across a number of categories in independent customer surveys. UCSD is a major world-class comprehensive university, one of the very best public universities in the United States, and a leading research university in engineering, biosciences, and biotechnology. The Jacobs School of Engineering was recently ranked among the top 10 institutions in the world by also an independent ranking. So that was the first objective, is to get tighter. The second one is to foster multidisciplinary thinking. Not one single discipline is going to attack some of the problems ahead of us, whether it's global warming or cancer, you name it, you know them all. And it's about bringing things together that things happen. Innovation happens when 2 ideas come together, and 2 ideas come together when 2 people come together. So multidisciplinary through science and technology discussions, looking for novel opportunities in emerging area. The third objective, which is actually an important one too, is we UCSD, we Invitrogen want to take a prominent stand in elevating the role of science in society by showcasing how society would benefit through cutting-edge science and technology. And finally, the fourth objective is we wanted to provide a unique educational opportunities for everybody involved here, starting with ourselves. So, to help us launch this shared vision, we are very, very pleased and excited to have Dr. Robert Langer from MIT to join us. The caliber of Dr. Langer's contribution to science and technology defines the high quality of this program. His topic for tonight, biomaterials and how they will change our lives, illustrates the type of multidisciplinary work we want to emphasize. My UCSD colleague, Xu Chen, will be introducing Dr. Langer in his lecture in a moment. Frontiers in Biotechnology will continue tomorrow with Professor Langer and a small group of invited UCSD and Invitrogen scientists who will participate in a professional development workshop to explore further the opportunities for transformative medical treatment. Following this evening's lecture, as Frieder mentioned it, there will be a dessert reception. Please join us to socialize with friends and colleagues and to continue the conversation about how together we can impact the future of medicine. I want to thank UCSD School of Engineering and Dean Frieder Seiblit for hosting Dr. Langer's seminar, and thank each of you for joining us tonight. And with this, it's my pleasure to introduce Dr. Xu Chen, who is a professor of bioengineering and who is also a university professor and one of the people who are member of the 3 illustrious academies, as Frieder mentioned it.
Thank you very much, Claude. Dean Seiblit, Vice President Benchmore, Vice Chancellor Brenner, Professor Langer, Professor Fung, ladies and gentlemen. At this auspicious occasion of the inauguration of the Invitrogen UCSD Frontiers in Biotechnology Distinguished Seminar Series, we are most fortunate to have Professor Robert Langer, who is the most distinguished leader in bioengineering and biotechnology, to be our inaugural keynote speaker. Dr. Langer received his bachelor degree from Cornell University and his Doctor of Science degree from MIT. After a postdoctoral training period, he went back to MIT to become a faculty member. He is a institute professor at MIT, which is one of the most distinguished and highest honor at the institute. Dr. Langer's groundbreaking and pace-setting work at the interface of bioengineering and biotechnology and material sciences is superb and unparalleled. He has developed novel methods for molecular delivery that allows the delivery of drugs with precise control of drug dosage, release rate, duration of action, and the targeting sites. His novel discoveries have broad implications. For example, in the treatment of cancer and many other diseases, in tissue engineering and regenerative medicine, the control of stem cell growth and proliferation, nanoscience and nanomedicine, and many others. He has really done so much to improve the human health and the quality of life. His innovation really has tremendous impact on all of us. Just like his title said, it changes our life in the positive way. As already mentioned, Dr. Langer is a member of all 3 U.S. academies, national academies, and he's also a fellow of the American Academy of Arts and Sciences. Because of his tremendous contributions in science and technology, he has received many awards and honors, which are too numerous to list here, but I'd like to mention 2. In 2002, he received the Charles Stark Draper Prize from the National Academy of Engineering, which is the highest honor for the Academy. Last year, he received the United States National Medal of Science, which is America's highest honor for scientific achievement. He and Dr. Fong are the only two bioengineers who have received the National Science Medal. Ladies and gentlemen, Dr. Langer is the best among best. He is an outstanding leader at the frontiers of biotechnology. Therefore, it's most fitting that he is the inaugural keynote speaker today. We're most fortunate to have Professor Langer here. Let's give him the warmest welcome to deliver his inaugural lecture.
Well, Dr. Shen and Dr. Fong and everyone, thank you. And I'm really very, very honored to be here tonight to give this lecture. When I was asked to give the lecture, one of the things that they suggested to me was that I perhaps focus on a particular aspect of biomaterials, and that would be nanotechnology. And so that's what I'll do tonight, is to try to focus in on that. But I thought what I'd start out doing is tell you a little bit, just autobiographical, about how I actually got into this field. And then what I'll do is go over some of the work, it's just some general comments about nanotechnology, then talk about new delivery systems based on nanotechnology for drugs, including even new ways of delivering DNA and siRNA. Move on from there and talk about new types of materials that we're working on, new kinds of microchips that might have nanowells to deliver drugs, and then new types of adhesives based on nanotechnology, and finally some new sensing devices and even ways of engineering tissues. Well, first, how did I get here today? So my background is chemical engineering, and when I was studying chemical engineering, my graduate work was at MIT in the early 1970s, 1970 to 1974. And for those of you that are old enough, I'm not sure what it was like in San Diego, but in Boston, what we always heard was that there was this enormous energy crisis in the 1970s. I'm not sure if it was real, But what you'd see, not unlike today, is that the prices would keep going up. But actually, in my opinion, at least in Boston area, it was much worse in the 1970s, because not only the prices keep going up, but if you had a car, you actually had to wait in line at the gas station for about 2 hours to get gas. And, time is, to wait 2 hours, that was really bad. But the consequence of that is that if you were a young chemical engineer, you got all kinds of job offers from the oil companies. And so I actually, all my friends, almost every one of my friends in my class at MIT went to an oil company or chemical company. And myself, I got 20 job offers in the different oil companies. 4 from Exxon alone. And it wasn't like my grades were that good or anything, but they just had so many openings. One of them in particular made quite an impression on me. I remember flying from Boston to Baton Rouge, Louisiana to Exxon. And when I was there, one of the engineers said to me, he said, if you could increase the yield of oil by 0.01%, he said, that would be wonderful. He said, that would be worth billions of dollars. And he said, isn't that terrific? And I remember flying back to Boston that night thinking, I just don't want to do that. So what would I do? This was what everybody did. So actually, I was very interested in education. I had when I was doing my thesis, one of the things I spent a lot of time on was helping a school for poor kids in Cambridge. And I developed new math curricula and chemistry curricula, and I really felt we made an impact. So I thought to myself, so one day in Chemical Engineering News, that's what chemical engineers read, there was this advertisement for developing chemistry curriculum at City College in New York. And I thought, this is great, I'm going to apply there and see what they'd say. And so I wrote them, and actually they never wrote me back. It was a little different than the oil companies. But I liked the idea. So then I started looking in these education journals like Chronicle for Higher Education and things like that for jobs where you might do curriculum development. And actually I applied to about 30 different universities to develop chemistry curriculum or math curriculum, basically education. And actually a lot of these schools weren't that good, but none of them wrote me back. So, what I begin to learn is that if you're not in this box, the education box, let's say, you not only you don't get hired, you don't get a response. So then I started thinking to myself, what else might I do? Well, I was always interested in using my education to help people. And another thing I thought about that I'd always been interested in was medicine. So I applied to a lot of hospitals. They didn't write me back either. Quite a few, actually. So, and medical schools, they didn't write back. But one of the postdocs in the lab where I was at MIT, he said to me, there's a surgeon at Harvard Medical School, a children's hospital, named Judah Folkman. He said, sometimes he takes unusual people. It's true. I don't think there was anything negative about Dr. Folkman, more about me. But so I wrote to Dr. Folkman, and he offered me a job. Didn't pay anywhere near what the oil companies did, but it was very exciting because he had an idea about that if you could stop blood vessels, you could stop tumors. And so my postdoctoral job was to see actually if I could isolate the very first molecule to stop angiogenesis, to stop blood vessel growth. Critical to that, solving that problem, was developing a bioassay to study that phenomena. And the bioassay that we wanted to use involved the eye of a rabbit. Because if you try to studying blood vessel growth is incredibly difficult because almost any part of the body that you look at, there's lots of blood vessels. The eye of the rabbit, there's no background blood vessels. So our idea was to put a tumor in the eye, like in the cornea. If you put the tumor in the cornea, blood vessels over a couple-month period grow from the edge of the cornea to the tumor. To stop that, what we thought is we'll put a slow-release polymer right next to it to take any of the molecules, large molecules that we were trying to isolate, and slowly deliver them over the several-month period to see if we could stop the blood vessels from growing. So that was actually how I got into doing this. And so my earliest work in the 1970s was to try to do two things. One, isolate this angiogenic inhibitor, and two, to develop controlled release polymers that were very tiny, that you could put in the eye of a rabbit to slowly emit whatever molecules we were testing. And that's what got me started. We were lucky we were able to do both those things. There was 2 papers, one in Science in 1976 on the isolation of the angiogenesis inhibitor, one in Nature and also in 1976 on the development of these controlled release polymers for macromolecules. Over the years, we continued to work on various aspects of biomaterials, creating different new materials for a whole range of things, cancer treatments, creating new tissues, and so forth. And one of the things that we also got involved in, I guess before it was fashionable, were nanoparticles. And that's sort of what or nanotechnology, and that's part of what I'll talk about tonight. In particular, the area of nanomedicine involves all types of things, and I'll try to give examples. Some I'll go into some detail, others I'll talk more just to give you a feel for, but it could involve diagnosis, monitoring, drug delivery, and even construction of human biological systems at the molecular level using engineered nanosystems. And one of the things, you hear about nanotechnology, I thought I'd just show you a picture to give you an idea of how small these particles are. So here's a human hair. Here's a human hair, and a nanometer is 1/1000th of a meter. It's about 1/10,000th the width of a human hair. So it's really small. Why is it important in medicine? Well, there's actually a range of reasons why it's important in medicine. One, if you want to inject something into the bloodstream, probably you don't want anything, the thing is when people talk about nanoparticles, or any particle, usually you hear them say, well, the particle size is 2 micrometers. That's actually probably not right. When people say that a particle is 2 micrometers, what that means is the mean of all the particles is 2 micrometers. There's going to be a distribution. And the problem is if you had a mean of 2 micrometers, some might be 10 or 20 or even 30 micrometers, and that may clog up the bloodstream. That would be real bad. So you want the particles very often to be very small, so there's no chance of clogging the bloodstream. A second reason why, and I'll talk about this in some detail soon, why you want particles to be small, is if you're trying to really deliver a drug to a specific cell, generally, other than macrophages, if you want the particles to be taken up by the cell, generally it's probably got to be 200 nanometers or less. And so that's probably one of the most key reasons for developing nanoparticles is so they can get into cells. But there are other things, too, that can happen. Also, if you can imagine, if you make something incredibly small, the surface area is enormous. And as I'll show you, that can lead to new types of diagnostics, new types of adhesives, and so forth. And finally, you can also create new surface patterns. And as I'll go over, those new surface patterns can actually be used in directing cells to grow, and as I'll go over, maybe even enabling you to create new tissues and organs. So those are some of the motivations. There are probably other motivations, too, why people are interested in nanotechnology. I mean, also, there's a lot of funding for it, too. The government's been putting a lot of funding for it, but there are real scientific reasons for doing it. And some of the areas of impact that I'll talk about are enhanced, or I'll talk about most of them, but you could do enhanced imaging, more sensitive diagnostics like a lab on a chip, targeted drug delivery where the drugs might go to specific cells. It might enable someday new therapies, like this is what I meant, like delivering DNA or maybe even siRNA. And finally, these ideas of nanostructured surfaces for tissue regeneration. First thing, though, that I thought I'd address before I go over any of that is there's all kinds of books that you could read, like Michael Crichton wrote a book on nanotechnology, and actually there was just a medical book I just read on nanotechnology from Michael Palmer. I don't know if people read him. He's in Boston. But basically, when you read these books, and you don't need to read the books, you could just look at the popular press, period, you might get the idea that nanotechnology is very unsafe, that it's a great way to, damage people. And there are huge concerns about it. Actually, I think that Michael Crichton's book in Prey, I think the nanoparticles went wild and started taking over the body. And actually in this Michael Palmer book that I just read, I think they were trying to control the president's mind in it. Of course, maybe some people might think that's good. I don't. But at any rate, so people might think it's not safe. So let me start out by addressing that. And the way I guess I thought I'd address it is that nanotechnology has actually been FDA approved, at least in some cases, for many years. There are technologies called liposomes. And a good example is doxorubicin in a liposome. And the idea is if you give doxorubicin directly, it might cause heart problems. But sometimes they put it in these little liposomes, and the liposomes don't dump it right away, so it doesn't go to the blood right away. So it circulates around in the bloodstream. The liposomes are about 100 nanometers mean diameter, and they're approved for treatment of ovarian cancer. Patients get an IV treatment about every 4 weeks, and Doxil takes the active agent doxorubicin. Like I say, the liposome is a fat bubble. It's really that simple. So this has been used now for about 11 or 12 years. No safety issues, approved by the FDA. Another example is AmBisome, another liposome. I picked these just because they're drug delivery systems that actually go into the human body. So if you wanted to think that it was dangerous, this would be at the top of your list. Here they put amphotericin in. This is an antifungal agent that might have problems if you gave it again immediately. But when you put it in the liposome, it works better. And this is also approved. It's also got a diameter of 100 nanometers. It's approved in the treatment of cryptococcal meningitis and HIV-infected patients and fungal infections. And it's also given IV. It's also been given to children. Again, it's a liposomal delivery system. So this is not to mean that all nanotechnology systems are going to be safe any more than any new drug or any new particle will be safe. But I think the idea that just because something's small doesn't necessarily mean it's bad. That's the only thing I'm really trying to illustrate. The idea that something's inherently small doesn't mean it's going to be unsafe. And in fact, the FDA has approved a number of these different systems. Okay, so with that as background, let me now go over some possible exciting things that one might do in drug delivery first. So one of the things that I think really would be everybody's dream is that if you could have a drug and have it just go right to the cell you want, no other cell in the body. And there's actually been some work on that, and it's exciting. There are a range of technologies. Some involve what I call naked ligands, where you might have an antibody or something like that, or an aptamer, and that in and of itself might be a drug. And there are a number of these that have been approved. There's also fusion proteins, radioimmunoconjugates, chemoimmunoconjugates, but if you look at any of these, when they get to their target, they're really very few molecules, right? None, 1, 1, 2 to 8. So what would be good about a nanoparticle? So the idea is if you could put a drug inside a nanoparticle, you might look at the nanoparticle as a little bag, a little fatty bubble, or a little polymer bubble. But if you could put the drug in a nanoparticle and have the nanoparticle go to the site you want, you can put enormous number of molecules in it. In other words, rather than just a few, you could put 1,000 molecules to 100,000 molecules in a single nanoparticle. So in a way, you know, like I'll just pick hockey as an example. You get a lot more shots on goal. So the chances are if you have a lot more shots on goal, you're going to get some hits. So by having nanoparticle with the drug in it, if you could in fact direct it to the site, like people are trying to do here, that might really open up new kinds of chemotherapy regimens. So that would be why it would be so good if this could be done. But as we can see, so far there's no approved drugs. And by the way, it's very challenging, so let me go over what the challenges are. If you want to make a nanoparticle, and people have tried to do this, there are a lot of things that you want to be able to do. You want to be able to encapsulate a reasonably large amount of drug, say over 30% by weight of the particle be drug. Also, you don't want to waste a lot of it. So generally, if you had to put it ideally, you'd like it to be greater than 80%. Also, you'd like to have the particles be able to be freeze-dried just because they'll be much more stable. You know, you could ship them around the world. You'd like them to not aggregate in the body. You'd like them most of the times to be degradable, so they won't accumulate in the body. You'd like them to be small. This is the point I mentioned of 200 nanometers or less, so they'll be taken up by a cell. And finally, and this has been one of the big challenges, you don't want them to be rapidly cleared. Let me just go over that. So if you give foreign particles in the body, what happens is the body is a natural scavenger system, macrophages, that eat foreign particles. So if you gave most particles into the body, they wouldn't get to where you want because the macrophages would eat them first. So how could you stop that? Well, a number of years ago, we actually published a paper in Science addressing this point. And what we did, and others have done related things, is we made a system where we had polymer made out of an FDA-approved polymer, lactic glycolic acid copolymer. We put the drug in it, and we coated it with polyethylene glycol, which is actually FDA-approved. The way we made these is we had two phases. We had an aqueous phase where we'd have water and a surfactant. We'd have an organic phase, which has the polyethylene glycol, and it could be anything, but we generally use polylactic glycolic acid because it's also FDA approved. We put that in organic solvent. You mix them together. The drug goes in here. Mix them up. We use ultrasound or microfluidization, and we get these what we'll call hairy nanoparticles. We dry off the solvent, and you get little nanospheres. Just to show you what they look like on atomic force microscopy, they're pretty spherical. And they actually have a pretty tight particle size range, centered in this case around 150 nanometers. So we were able to do that. Now, one of the things that I mentioned to you is that probably the biggest challenge when we looked at that list of 7 is making sure they're not taken up by macrophages. So we actually developed a simple in vitro test to do that. We took macrophages and we put an orange dye in the nanoparticles. So if you have the polymer nanoparticles without polyethylene glycol, each of these little dots that you see in the macrophage, this is actually an hour after we incubated them, but you see tons of little orange particles in the macrophage. So the macrophage ate them pretty quickly. But if we put a 5,000 molecular weight peg chain on, very, very few nanoparticles are taken up. 20,000, also very few. And we even developed a chemistry using citric acid where you can actually have 3 peg chains and you get really good surface coverage. There, I think if you look on the bottom panel, this bottom panel, you really can't see any particles taken up by the macrophage. So by doing this peg coating, we were able to make it really difficult for the macrophages to eat these little particles. Going in vivo, we see the same thing. If you look at the dose, as you increase the polyethylene glycol molecular weight, we were able to affect the clearance, make it slower and slower. So coating with PEG is a way. PEG is extremely hydrophilic. Makes it extremely difficult for the macrophages to eat things right away. So how could we now get to the cells we want? Well, about 6 years ago, Omid Farrokhzad, he came to my lab as a fellow. Now he's assistant professor at Harvard. What he decided to do was work on making these nanoparticles not only have PEG on them, which is this, but have a targeting molecule on, which are these arrows. This is the kind of artwork we do. And you could use different molecules, right? You could use an antibody, you could use an aptamer, that's a piece of RNA, or we've actually developed screening systems for different small molecules. One of the key challenges, in fact, we just published a paper on this recently in PNAS, is getting exactly the right density of targeting molecules in PEG, because it's a real optimization problem. If you have too much targeting molecule, well, that gets rid of the PEG effect, and then the macrophages will still eat them. And if you have too much PEG, well, then you don't get to the cell you want. But you can actually do an optimization and get the right amount. And when you do, this was also published in PNAS, you could get some quite exciting results. So basically, let me just show you that. So the idea is that here what we did is we took a tumor. This is prostate cancer tumor in mice, nude mice, and you grow it up to about 300 millimeter cubed. And then you could do one of three things: a control nanoparticle, Taxol by itself, or these targeted nanoparticles. And what you see is actually, we were very excited. This was quite remarkable. Whereas the controls all grow, the treated animals shrink to almost nothing. Here's another way of looking at it. This is one of these animals. Here's the tumor, maybe 40 days out. Here's what it looks like excised. Here's another control with the drug itself. Again, you see the big tumor mass. Here you see it excised. Very, very vascular. Yet, when you use the targeted nanoparticles, pretty much goes away. And here's what the tumor looks like. I mean, there's basically no tumor. It's just white tissue. So the hope is that this could provide a whole new strategy for getting drug targeting with nanoparticles. And our hope is that we actually started one of the things we do at MIT, like you do here, is we start companies to develop the manufacturing and stuff. So there's a company called Bind that Omid and others started. And so the hope is that this will be in clinical trials next year. So we'll see how we do. But that's one example. The second example that I wanted to talk about with nanotechnology, and again, all these things I'm talking about are pretty early stage, but the second example that I wanted to talk about is actually delivery of DNA. And this has been a huge problem. And let me illustrate that, how big a problem it is and what the challenge is by giving you a quote from a very famous person from San Diego. And that's Inder Verma, when he was president of the Gene Therapy Society. This is actually from Time magazine. We look at all kinds of journals. When he was asked what was the biggest problem in drug delivery, in gene therapy, he said, well, there are only three problems in gene therapy: delivery, delivery, and delivery. That was 9 years ago. And in fact, a lot of my students quote this. It's kind of funny when you look at references in a thesis, and you see Science, Nature, and then Time magazine. But it actually is a pretty good quote. The problems, and people I'm sure know this, have been safety and efficacy. And of course, by far the prime approach that people have used have been viral vectors, like adenoviral vectors or retroviral vectors. But again, I'm sure people are all aware that there have been problems when people have done that. Safety problems, sometimes people have died. So another approach then might be synthetic vectors. They're potentially safer, cheaper, and easier to manufacture. The problem is they work very badly, at least up till now. They're very inefficient. And so it's been hard to develop a synthetic vector that's been working real well. I was fortunate about 10 years ago. I got a call from Bob Grubbs, who's a chemistry professor at Caltech. Bob actually would receive the Nobel Prize in 2005 for coming up with chemical reactions, including polymerizations, where he was able to reduce the number of steps. This was part of what he got it for, from about 15 to 5. And Bob called me up in about 1996, 97, and he said his number one graduate student, David Lynn, wanted to join our lab, and would we take him? So Dave came to the lab, and one of the things that he began doing was thinking about synthesis approaches to make polymers. But I should point out that, and as I mentioned in my introductory remarks, we make polymers a lot in our lab, new biomaterials. But whenever we do, it probably takes us about 4 or 5 months. It's not that we're bad, but the problem is when we make new polymers, the way you do it is you start out with what's called a monomer, small molecule. And of course, there's all these functional groups on the monomer, so you have to block some. Then you do a reaction. Then you have to purify it. Then you have to unblock the reaction. Then you have to do your next step, and you have to block it again, purify it again, unblock it again. By the time you're done, your yields are low. It takes 20, 30, 40 steps, and many months have gone by. So what David wondered about is, could he come up with a chemistry whereby he could do it in a single step? And what he recognized was that there was a particular chemistry if he would just use what's called a Michael addition of amines to diacrylates, amine here, diacrylate here, that the one-to-one correspondence of this to this would mean no side reactions. So you could simply add these like this or like this, and with different R groups, you'd have enormous diversity. So the possible advantages of this kind of chemistry, and again, it's limited to just this chemistry, you couldn't do it, are that you could use commercially availing starting materials with lots of different R groups. But the big advantage would be that the polymerization could be accomplished in a single step, without byproducts, and without protection or deprotection. So rather than taking months to make a single polymer, what we thought is that we could do high throughput and really, literally make thousands of polymers, hopefully in a few days or a few weeks. And so Dave, working with Dan Anderson, I'm doing high throughput too fast. Let me just, I'm not sure why it's not staying there. Let me just, do we have somebody who's expert at projectionist? What's that? Is there somebody who's good at, what? Yeah, well, actually, I'll do it very. I'm going to go back to the last slide, but you won't be able to see it very. This is really high throughput. But what I was going to show you on that last slide, which for whatever reason is going by really quickly, is a study that Dave Lynn and Dan Anderson did. And what they literally did is they took something like 94 different aminopolymers, and since the slide's going to go quickly, all I really want you to see is the enormous chemical diversity of those 94 aminopolymers. And then they took 25 acrylate monomers, and they simply mixed each of those 94 separately with each of those 25. So you get over 2,000 different polymers with very different chemical structures. So let me go back, and hopefully you'll see it real quickly. So on the top, you're going to see the 94 amino monomers. Is there a way to do it?
Just take it back to the take out of the show.
See, I'm okay on chemical engineering. When it comes to computers, I'm not so good.
Can you take out of the slideshow?
I'm not sure.
Take it out of the slideshow.
Oh, okay. So how do
I'll put it back.
okay, so you can go to the last. Oh, perfect. Okay, so at least I'm very lucky to be in good company here. You know, you don't get into these 3 national academies for nothing. Well, he'll get in next, then, right? So anyhow, but at least Shu was smart enough to think of that. I wasn't. So at any rate, so basically. So here are the 90. So what I really wanted you to see are the 94 amino monomers, and the whole point is the enormous chemical diversity. 25 diacrylate monomers, again, enormous diversity. So basically, we had robots do a lot of this, and what we made, you take each of these with each of these, so you get 2,350 totally different polymers. Oh, now we go back to this. Okay, but sorry that was so long. But it's just to make a simple point that you get enormous structural diversity. And what we found, and we even made secondary library, we made more than the 2,000. You can purify out different molecular weights and things like that. But there are a lot of different ones. And the top 5 are shown here. This is actually the best one. We call it C32. But they're all more effective than lipofectamine 2000. They all have alcohol side chains. But we did over 25,000. Again, they developed robotic methods for doing the transfections, shown here using a luciferase assay. And I thought I'd just take you through 1 or 2 examples of what you could do with it. So one, Janet Sawicki, who was at Penn and approached us because she wanted to deliver the genes for diphtheria toxin, see if we could affect prostate cancer. And again, by going through the screening, what you find is buffer has not much effect, DNA has small effect, polyethylenamine, one of the standard things for gene therapy that people use non-virally, has a little effect, but C32, pretty substantial effect in terms of luciferase. So you can actually light up the prostate tumor with the C32, and you're much, much better than anything else. In contrast, C32, let's take a look at normal tissue. So with what you see with C32, with an IM injection, you get the lowest expression with C32. So it seems to be pretty specific for the tumor. So basically, it's pretty ideal for local delivery of tumors near and in muscle. So we have to also ask, is it safe? Again, we've done a lot of cell-based screens, but in vivo is more important. So one of the ways to look at if it's safe is to do histology. So here's polyethylenamine, and here's polyethylenamine in the tumor. By the way histology works is that you take an animal, you inject things in it, you take the tissue out, you do what's called fixing it, like there's different substances like glutaraldehyde, and then you do a stain with some dyes like hematoxylin and eosin. Now, if you're a really good pathologist, you could look at it and know whether it's cancerous, inflammatory, or anything else. If you're somebody like me, what you do is you look for purple dots. And the way I would tell if something is inflammatory is are there a lot of purple dots or are there a few purple dots? This is what I'd learned from pathologists. And PEI is pretty biocompatible. You can see there's not very many purple dots. There's some, but C32, even though there's 30 times more in this study, there's almost no purple dots. So this is actually quite safe in terms of tissues, and there's, like I say, 30 times more. And here's an early study that they did where basically they were able to get a 40% regression in just 2 weeks with an intratumoral injection. So that was exciting. And Dan and Janet are actually recently published a paper in prostate showing actually very good treatment, not only for this, but they've done some studies on ovarian cancer and hope to take that into the clinic in the next few years. But then what some of our students, Jordan Green and others, did is actually take the library we had, which already worked pretty well, but then modified the N groups. In other words, and so they made an additional library where they took the best of the over 2,000 polymers, made another several thousand polymers, and they got some that worked far, far better. And here's one of the things that we just recently did, which is actually very exciting to us, is that we actually compared, these are cells using fluorescent reporter gene, but here's polyethylenamine, here's actually adenoviral vectors. And you can see by far the best effects, at least after 24 hours. This is cell-based. This is not in vivo yet. We hope to take it in vivo. But you get better, but I think the exciting result is that you get better transfection with the polymer vector than you do with either the adenoviral vector at a high multiplicity of infection or the existing polymer material. Another thing that we've been working on, I don't know how many people have followed some of these stem cell things, but you may note that recently, there have been studies where Rudy Janish, who's one of our collaborators, and others have been able to take 4 genes, and by giving 4 genes, with a viral vector, they've been able to take a normal cell, like a skin cell or a fibroblast, and turn it into a stem cell. So one of the things that we want to do, and we're collaborating with him, is could we take a polymer vector, take the 4 genes, and take a normal cell and convert it to a stem cell. If we could do that, someday that could provide a whole new way, and a safe way, because the viral vectors are not safe, of doing this. So as an early demonstration, what we've done is simply to almost do the reverse. Could we, but we have a lot of studies going on the stem cell things, but here we've actually transfected with C32, that's the polymer we used before, and this is with certain N groups. And basically, we were able to make the stem cells light up with this fluorescent assay, whereas, again, with the standard molecules, you're not able to. So we hope, with further work, using this kind of nanotechnology, that we'll be able to deliver genes someday. Again, very early, but it's the kind of thing that nanotechnology holds promise for. Now, by delivering genes, by delivering DNA, you can sort of give a cell a function that it didn't have before, right? But there's a whole other thing that you might like to do, and that's where siRNA comes in. The first idea might be to turn genes on, but the second idea is could we turn genes off? And in 2006, the Nobel Prize was given in medicine and physiology for exactly that, for the discovery of fire and mellow of siRNA. But again, just like DNA, the problem with siRNA, trying to turn genes off, is delivery. People have not been able to get siRNA into cells in a safe and effective way. That's probably been the single biggest problem. So what I'm going to tell you now is actually going to come out, and it's actually the cover story of Nature Biotechnology, I think in 2 weeks, maybe next week. But basically what we did here is use a similar strategy. Again, high throughput. So one of the things that's very useful in delivering molecules, as I mentioned earlier, are liposomes, or at least lipid vehicles. So one of the questions that Dan Anderson in our group asked is, could we make lipid-like materials that might be useful for delivering siRNA? Now, let me point out the following, and people may know this if you follow the liposome field. But liposomes have been around now for 43 years. They've been very useful in a variety of things and actually been approved by the FDA. They're certainly used in biology as well. But if you looked at the liposome field, I would say that probably almost all liposomes are based on about 10 different lipids. And if you looked at the entire literature going back to 1965, maybe you'd see 50. So again, we thought, using these high throughput approaches, again, maybe we could change the diversity. Maybe we could make hundreds, thousands, and find some that might be really good to solve this problem. So again, what we did is we decided to use this kind of chemistry to take the acrylate and the amines and develop a high throughput approach to make lipid-like materials shown here. And let me just show you the diversity. So first we could get, again, large variation in R groups shown here. And also what we could do is change the number of tails. Like, in other words, this is like a 10-chain carbon, 14-chain carbon, 18-chain carbon, and so forth. You could also put amines on it. Not only could you change the length of the tails, you could change the number. This is 5 tails, right? This is 4. This is 3. So again, you could build an enormous chemical diversity. Different R groups, different number of tails, different lengths of tails. So rather than just take 10 lipids, you'd have the ability to make thousands, tens of thousands. Maybe we could find some that are really good. So what we did is we actually made a bunch of these. And we actually, I don't think it's probably fair to say that they're exactly like a liposome, because the structure is not quite the same. The way we mix them together in similar ways, but they don't have exactly the same structure as a liposome. They sort of, when you look at them under like electron micrograph, it seems like they kind of surround the siRNA. But anyhow, the important thing is that we found we used the following type of assay. Again, we developed a high throughput screen. We took a luciferase assay, a cell-based assay, and the idea is we actually looked at 2 types of luciferases, Firefly and Rhinella. And if you knock them both down, that means it's probably toxic. But if you just knock down the one you're trying to knock down, say Firefly, that means it's effective. So that's what we did. We screened thousands, and we find actually 56 that work as good or better than lipofectamine 2000. Some of the key chemical features are amide linkages, more than 2 alkyl tails, and a tail length in the range of 8 to 12 carbons. So then we went in vivo. This, by the way, is all being done in collaboration with Alnylam, which is a local company in Cambridge, which I'm on the scientific advisory board of. And they've made all of the siRNAs. In fact, one of my students works there and did a lot, who's the first author on the Nature Biotech paper. But basically, what we would do here is make a siRNA against factor 7. And what you could see is that, or against ApoB. And what you can see is that if you make it against factor 7, you get about what we call 80% knockdown. Here's another siRNA, I'm sorry, another different type of lipid, and again about 80% knockdown. And then if you change the siRNA from factor 7 to one against ApoB, here you get about 65% knockdown for the ApoB. So you can make them very, very specific. So that's in animals. Also, what's really exciting is a single injection. You get an effect almost for a month. So this is small animals. And you get a dose-response curve. Here's phosphate buffer saline. Here's the control siRNA. Again, this is all factor 7. And you see as you increase the amount from 1.25 to 2.5 to 5 to 10, you're able to get more and more knockdown, almost complete knockdown, when you go to 10 mg per kg. And no safety problems to the animals. So then what we've done very recently in that paper in Nature Biotech is to actually go to monkeys, to go to primates, to see if we could get this ready for man. And here we're looking at ApoB again, ApoB, as I'll go over in a minute, has to do with cholesterol. And here again, a single injection is able to get some effect in the monkeys. The standard deviations are significant, but some effect over a 30-day period. And here we're going to be looking at are we able to knock down the protein level. Again, if we're looking at liver ApoB, here we see control 2.5 mg per kg, 6.25 mg per kg, probably about 85% knockdown. That translates now to the serum ApoB level. Again, after 48 hours, you get very significant knockdown at a 6.25 mg per kg dose. Again, this is all monkey data. And finally, the clinical manifestation of that might be, could you lower cholesterol? And here you see your LDL cholesterol lowered nearly about a factor of 2, so about 50% lowering. This is 48 hours later. So your LDL cholesterol, when you give the siRNA against ApoB, is lowered by 50%. So if you had, for example, a cholesterol or an LDL cholesterol of 200, that would mean now when you give this, it's 100. So basically, that could someday be a treatment for hypercholesterolemia, but I think what everybody's more immediately interested in is trying to target it to liver diseases like liver cancer, other things like that. So this is another example of nanotechnology. And what's really important about it, all these complexes, again, are about 150, 100 nanometers. And what's really important about it is if you didn't have these kinds of approaches, and there's others that people are working on too, you wouldn't be able to use the siRNA. You wouldn't be able to solve the delivery problem. Well, let me go over a few other things and again, I'll go over all of these a little bit more briefly, that you might be able to do with nanotechnology. Here, this is a little bit different, the next example, but it's kind of microfabrication. And about 15 years ago, I happened to be watching a television show on PBS. And this particular TV show, they were talking about how microchips are made in the computer industry. And when I was watching that TV show, I thought to myself, wouldn't this be a great way to make a drug delivery system? Now, maybe if you spent 30 years of your life just working on drug delivery systems, any TV show you saw, you might think that. So it's not that I was that creative. But when I saw it, I thought this was really interesting. What if we could make a chemistry chip, a drug delivery chip? So I contacted Michael Sema. He's one of my friends in material science, and he's one of the top people in the world in ceramics processing. And here's what we started talking about. Could we make a little chip? And in this chip, we're building wells. And each of these wells, you could put different doses of a drug, or you could put different drugs. You could literally, if the drugs are potent enough, have a pharmacy on a chip. In other words, all the drugs you'd ever need, and they could come out whenever you want. So this was the idea, that we would make these chips. And they're all using, and again, I don't know how many people, or if anybody really is in the pharmaceutical industry, but classically pharmaceutical processing uses a variety of approaches. And here what we're using are approaches based on, that are really used much more in the microprocessing industry, like reactive ion etching, photolithography, and to make little tiny wells that are nanometer scale. Or micrometer scale. And basically, the idea is we'd make these wells, put the drugs in them. We actually do this by either inkjet printing or computer-aided microinjection. And in each of these wells, we would put, like I say, different doses of the same drug or different doses or different drugs. Let me just show you a picture of an early chip that John Santini, one of our graduate students, made. Here's a top view with 34 wells. Here's a bottom view with 34 wells. Here's a United States dime. Now, this chip is kind of like a regular chip, but it's kind of like a flat chip. But in our laboratory, we have students who've made all kinds of chips. We have actually made tiny, very, very ultra-thin cylindrical chips that you could inject through a trocar. So they're actually injectable, like the NOR plant. We've made chips that are totally degradable. All kinds of chips. That's good thesis kind of stuff. And let me just show you how they work. So here we're looking at a single well covered with gold. Gold is one of the molecules, the substances we use. And gold is actually very good. It's actually quite biocompatible, it turns out, in the body. And when you put it in the body, it actually doesn't corrode at all. We looked at a variety of different metals. It'll actually stay like that for years. But it turns out if you just apply 1 volt in the presence of a small amount of chloride, here's what happens in just 10 seconds. The gold dissolves. It forms a soluble complex. And when it does, whatever's underneath it could come right out. So it's kept in place as long as you want it to be, but then immediately triggered when you apply this external voltage. And one of the things we've done is, we haven't tested this in patients yet either, but we've collaborated here very closely with Jim Anderson. He's one of the top biomaterials pathologists in the world. He's at Case Western. He's found that if you take 16,000 times this amount of gold, you don't see any harm to the animals. So basically, that's what you can do. And here's just a study that John Santini, one of our students who really led this work, did. He's triggered release of different amounts of drug in different wells over a 6-day period. And again, these are just pulses that he's gotten. That's a single drug. Here he's done more like the pharmacy on the chip idea. And he's triggered release of one drug at 25 hours, another at 37, this same drug but from a different well at 50, and this same drug from a different well at 60 hours. In fact, John also started a company on this technology called Microchips. It's in Bedford, Massachusetts. And about 1.5 years ago, he and the people who worked there published a very exciting paper in Nature Biotechnology. What they did is they put these chips in dogs, and they put a peptide in it, which is a pretty big molecule, and then by telemetry, the same way you'd open up a garage door, they just triggered release over and over again. And they got the same amount at day 1 that they did at day 10, that they did at 1 month, that they did at 3 months, and that they did at 6 months. So so whatever amount, one of the concerns people might have is, do these get encapsulated? Does that change release? Well, the answer is, at least with a peptide, nothing so significant. I mean, there's tiny encapsulation, but nothing so significant that it even changes the release rate. And also what people are now trying to do, they're not only trying to use these chips for delivery, but also for sensing. People might realize that when you put a sensor in the body, like say for insulin, or for glucose rather, or for other things, one of the biggest problems with the sensor once it's implanted in the body is that it fouls. So one of the things that they're doing is they're actually putting sensors into these chips, and every time one fouls, they just open up another well, has another sensor. So the idea is that you could make incredibly long-acting sensors. And so they've been working with Medtronic and other places on that. And then someday, and again, this is all speculation, but someday we'd like to believe and hope that you could actually take those two pieces and put them together, that you could have a delivery system that could sense what the body needs, and I'll even give you another example shortly, and then whenever it needs something, the chip then tells it how much to deliver, so you could actually have a closed loop. Okay, so that's the idea of the microchip. So I thought I'd also try to tell you about some new nanomaterials that we're working on. And this is an idea, the next one, that, again, is kind of more what I'll call still at the science stage. It's not like I have a specific application, though, so we can have a test to see if people can think of them here. What I'm going to talk about is not only just nanotechnology, but even what you might even consider picotechnology. That's an order of magnitude less. So here, Jörg Lahan, and this is actually something we published in Science, so it's decent science, but we're still thinking about what's the key killer app. So the question is, really, can we manipulate surface properties of materials? So the idea that we had here is, if you had, let's say you had a material, and let's say we had molecules. We might even pick self-assembled monolayers, but let's say we have molecules, and let's say you've got a surface, and now we build these molecules on top of these surfaces, and they'll stand up. And I'll just pick a simple example. Let's say I just want to change the surface from hydrophilic, in other words, water-loving, to hydrophobic, water-hating. Could I do that? Could I throw a simple switch and just change the surface, like have a table, go from hydrophobic to hydrophilic, whatever you want? So here, let's say we build in a set of molecules. This is just theoretical for now. We build in a set of molecules that are very, very hydrophilic. With the molecule has a red part that's very hydrophilic, and also we'll build in a little negative charge. Let's say on the same molecule, I make the center part of it, though, very hydrophobic. Okay, so initially the molecule looks like this. But if I put a negative charge on the surface, what if I just throw a switch and change the surface and now make it positive? Well, what we would expect, perhaps, is that these negative charged molecules, when the surface is negative, will stand up. But when it's positive, they'll turn over. So we've actually changed the surface from hydrophilic, this red part, to hydrophobic, this part. This is purely theoretical. This was the idea that Jorg Lahan, myself, and Samir Mitragatri had. Could we do this? Well, not so easy. But Jorg synthesized self-assembled monolayers. And then what he did is he put on, and we tried to do this. And the problem is whenever we try to do it, and we tried to turn over the molecules, it didn't work. And let me give you an analogy about why it didn't work. Whenever you synthesize these molecules, envision like you have a forest, okay, of trees, and let's say we cut the bottoms of those trees off. Well, the problem is if all the trees are very close together, they can't fall over, right, because you've got another tree right next to it. And that's the problem we had. So what Jorg and Samir did then is a lot of molecular modeling to figure out what's the right distance the trees or the molecules have to be apart. So they would fall over, and fall over just the right way. And they found out that it had to be a pretty good distance apart. So what they did is they built really bulky molecules. Here's got 3 phenyl groups, really bulky molecules. And the idea would be that they have these really bulky molecules, but then, and you use those bulky molecules to keep everything apart, but then when we want to get this to work, we simply chop off the heads of these molecules. You might think of them as mushroom caps. We forced the spacing to be far apart by making these very, very bulky. Now we take them off, and when we take them off, the spacing we feel would be good enough so now these can turn over. In fact, we've even done a simulation. So what I'm going to do is show you a simulation, molecularly what happens when they have this spacing. So this starts out negatively, but let's turn on the switch. So it's positive. It's standing up. Now we're going to switch it back to negative. Snaps right back. And we would say you should be able to do that over and over again. How could you prove that? You know, one of the nice things about giving lectures is that when you give lectures, you know, you meet interesting people and they show you their techniques. So I had to give these lectures at Berkeley Chemistry Department a number of years ago, about 4 years ago. And when I was lecturing there, one of the people who took me around is Gabor Summerjai. Gabor just actually won the Priestley Medal. He's one of the world's top chemists. And he started showing me some of the techniques he was using to analyze surfaces. And so when we were studying this, I mentioned this to Jorg, and he flew out there. And the technique that Gabor has pioneered is some frequency generation. It's a combination of various spectroscopy techniques. And the thing is that what you could see is that when you have if you start out like this and throw the switch on, you get a huge change in the SFG signal. Off goes right back. On, back again. Off, keep doing it over and over again. We actually did this 20 times. I'm not going to show you the whole 20. But basically, totally reproducible, goes back each time to where it was. Yet if you do controls, no effect. So you could actually look at the molecular level using these techniques and see that you could change the height, that you could change these molecules. By the way, I should tell you why I'm even calling it pico. So if the molecules are straight up, like I showed you in the earlier slide, that's about 1 nanometer. When they're bent over, it's actually less than a nanometer. So and so you're changing the surface over and over again, just like I'm changing my finger. The other thing that we did is just some simple measurements. From hydrophilic to hydrophobic, well, you could just look at contact angle. And here, what you see is with the experiments, you're able to change the contact angle. Again, the effects aren't as big as the molecular changes, but substantial. On, off, controls. Here's another control. So again, so actually my quiz for tonight, if anybody has ideas, what would be a good application for this? Again, usually we're kind of application driven. Here we just had this basic idea of how we could change molecular conformations and totally have a switch that could change the properties. And by the way, it doesn't have to be just hydrophilic to hydrophobic, right? It could be affinity for one molecule versus affinity for another. You could build these molecules, but that would be the basic principle. So I thought I'd talk to you about 2 or 3 other things to end the talk. All these are very, very preliminary, but I think they're interesting, so I thought I would just share some ideas with you. So This next one, Jeff Karp actually led in our laboratory. Jeff's now a professor at Harvard. This actually came out in PNAS a month or two ago, got quite a bit of publicity. But what Jeff and I were talking about was maybe making better adhesives. And what Jeff started thinking about is that there's a small animal called a gecko. And if you've ever looked at the gecko, it's got these feet, right? It's got all these nanopillars. So what he decided to do was to, again, use microfabrication. Again, take silicone wafers and etch them, and you probably have to look real closely at it. But here's the, and try to actually mimic a gecko. We also in our laboratory, as I mentioned, synthesized different polymers. One of the ones that Yadong Wang synthesized was something we call biorubber. It's also called polyglycerol subbasic acid. But it's one of the very few polymers that's very elastic, degradable, and biocompatible. So what we thought is that if we could also build in this adhesive property to it by this gecko structure, might really become a very powerful adhesive. Where might you use these things? Well, what kinds of things that you might think of is like, say somebody's doing one of these gastric bypass operations, you know, where you try to suture things in. There's all kinds of leaks. Another application some students were just talking to me about 2 days ago at MIT was lung surgeries, where, again, there's a lot of leaks. Could you have some simple things that you could work on and see? The problem with some of the sealants people are trying to use is they're just not strong enough. So, again, this is what, so what Jeff did is actually made some of these. This is an example of what he made. Here's it after 8 days. It still shows some of that structure. And they're basically like little pieces of rubber that you could just seal on in an operation, you know, for different uses. And again, there are probably all kinds of uses for this because it's just a very simple adhesive. The beauty of the gecko, the nanotechnology, you might recall in one of the early slides I showed you, is nanotechnology, you get enormous amount of surface area. So what that means from an adhesive standpoint is you've got so much more hydrogen bonding and van der Waals forces because there's all these tiny little structures smack up against the tissue that you want it to be. Another thing and again, this is still sort of at the idea stage that we thought about, was new kinds of implants someday that might be able to help in sensing. One of the things that we actually did at MIT is we were fortunate, and they have one here at San Diego too, but there were like 8 centers that were able to receive funding from the National Cancer Institute, getting $20 million to use nanotechnology in cancer. My co-principal investigator in this is Ralph Weissleiter at Mass General. And one of the things he's developed are these new MRI nanoparticles, which have metal cores, and he's actually been using them in imaging. He's developing new MRI particles that can be used in imaging because of their surface area. But what Michael Sema and Ralph and myself started talking, and Lee Josephson started talking about, are maybe we can do some other things. This is going to be in vitro. What if we take these little metal cores, put a polymer coating on them, and put a binding moiety on them? Well, you'll get since they're MRI particles, you could actually put them like in an MRI machine, or you could do an external MR, and you could actually get a T2 signal. That's the signal you get from MR. But let's say the binding moiety is going to, let's say it normally looks like this, but now all of a sudden you have an analyte that causes these to bind. For example, let's say these are all antibodies and this is the antigen. Well, this could all cause this binding. So if you go from this to this, let's say there's an analyte that you want to measure, and that analyte is specific for the binding moiety, which is easy to do, right? Pick the right antibody. You could make very, very specific nanoparticles for different analytes. And your T2 signal here is going to be very different from your T2 signal here. So this is the idea. So what Michael Sema and some of my students did is actually make a different kind of version of a microchip, where you could put in the little chip, this is like almost an injectable one, have a semi-permeable membrane here where any analyte, now we're going to go in vivo, could diffuse in from the body. And now you have these magnetic resonance switches in here, these MRS particles. They can't get out because they're still too big, whereas the little molecules, the analytes, can get in. So the idea is that we could implant this in animals. So what Mike and some of our students did is they picked a model, they put a tumor in, and then they looked at the HCG level over time. Just as an example, they would draw blood, and this is a human chorionic gonadotrophin B chain, and basically you can see that when you put the tumor in, the tumor actually is you get more of this molecule HCG being produced. So what we wanted to see is, could we someday have a non-invasive monitor, right? Could you have an implant in, rather than draw blood, could the patient follow cancer progression just by itself? Could you just go to an MR machine? And we're just picking this one molecule. You could imagine that when I showed you the last slide, you could have put 10, 20, 30 different sensors in, so you could get a whole way of following the cancer progression. But here's just an example of what we recently did. If you put a tumor in and you look at the T2, here's no tumor present, here's a 2T signal, and if the tumor is present, you get a big drop in T2, statistically significant. Again, here's the control, and what's done here is nanoparticles that are function for HCG that will bind to it, yes, yes, no, no. So you can see the only time when you get a change in T2 is when you have the nanoparticles functioning to the HCG and the tumor's present. So this is a proof of principle that someday, this is a week after the tumor is implanted, so this could provide a whole new way someday of using nanotechnology, these little magnetic resonance switches, to really monitor different features of what's going on in the body, and it might suggest to the patient or clinician what types of treatments to use. Well, the very last thing that I wanted to talk about today is, could we use nanotechnology to help create new tissues or organs? About 25 years ago, Jay Vacanti, who's a surgeon, he used to be at Boston Children's Hospital. Now he's head of pediatric surgery at Mass General. He's a very close friend of mine. And he talked to me about this problem of tissue engineering. Well, actually, what he talked to me about was, he said, Bob, you know, I'm treating all these little babies that are dying of liver failure. That's what he was doing in the early 80s. He was having a liver transplant program at Children's Hospital. And he said, you know, almost all these babies die. The only way I can treat any of these babies who are dying of liver failure is somebody else dies and I do a transplant. And he said, is there any way that we could develop a strategy, a way of treating patients with liver failure or other diseases by using their own cells or close relative cells? So he and I came up with this idea, which is one of the ways that people now try to create new tissues or organs. And the idea is that if you took isolated dissociated cells and inject them into the body at random, not much happens. But if you take those same cells, put them close enough together, they're actually smart. They can actually reform structures. There was a group at Berkeley that did some very nice work where they took mammary epithelial cells, put them in vitro, and they actually lined up and made acini and made milk. So one of the things that Jay and I thought about was, what if we could make polymer structures that were three-dimensional with the right architecture, put the cells close enough together, could they reform structures? We actually have to figure out the right way to grow them in vitro, which is also a challenge, to make virtually any tissue in the body. And I thought I would just give you two examples of what we've tried to do. Again, a lot of what we've done in this does not involve nanotechnology, and I might go over some more of that in tomorrow's sessions. But I thought I'd give you two nanotechnology examples. One was something that Chris Bettinger in our lab recently did. And the issue here is that for so many of these tissues, like when you're trying to build a new liver or anything else in the body, you need a vascular supply. How do you get this vascular supply? So what we did is we might take endothelial cells, or in this case, endothelial progenitor cells, that we did with Sharon Garrett, who is another one of our students, now at Johns Hopkins. And if you just took the cells and put them on a regular surface, they don't do very much. But if you create a structure which is nanostructured, like Chris Bettinger made all these, again using photolithography and etching, made these little templates with all these grooves, and then he lined them with these endothelial progenitor cells, and that helped guide them. So they'd make these little vessels, and they'd make really long vessels in some cases, which is very, very helpful. You don't see that in the regular structure. And so if you analyze it, the average length is much greater in the nano case than in the flat case. And this is just another example showing you that in the nano case you get some really, really long vessels, whereas in the flat case the vessels are very, very tiny. So we're beginning to use these kinds of approaches. Like, so you might just give you an example. Let's say someday, we've been working on this with Jay, we wanted to give somebody a new liver. So we wanted to put hepatocytes on a scaffold. But how do we get the blood supply in? So what we'd build in is a nano architecture where we could line them with these endothelial progenitor cells, they could actually vascularize the tissue. If you didn't do that, the problem is the liver cells would die. They don't get the nutrition or get rid of waste. But when they are lined with the blood vessels will help them get the nutrients and get rid of waste. So that's one thing that we're working on. Another thing that we're working on, actually, one of the people who we started, and this is actually, I'm going to nearly end the talk, is actually for spinal cord repair. Here, we've worked very closely with Evan Snyder. Evan is one of the top people in the world in neuronal stem cells. He's actually now at the Burnham. But when I first met him, he was at Harvard Medical School. And he's pioneered all this work with neuronal stem cells. So Erin Levick, who is one of my graduate students, she's now professor at Yale, began to work with Evan and Ted Tang, who was a fellow with Evan. And the idea that we had is, could we create a scaffold to help guide the cells? And we'd have an inner portion with large pores that would be seeded with neuronal stem cells, and an outer portion that I'll show you in a second that might provide axonal guidance. And so what was done is this is what it looks like. This is the inner part with the pores, but here's the outer part with these grooves, almost at a nanoscale, to help guide the axonal growth. It's kind of like you're making the inside like a gray matter and the outside like white matter. So the idea in these experiments is that, could we actually someday make people walk like Christopher Reeve or others, if we got them soon enough, by making a tissue-engineered spinal cord, again using this kind of nanotopology. So what was done is we took about 60 animals, all those 60 animals, we made an injury in the spinal cord which would make them paraplegic. In 15 of them, we gave cells by themselves as a control. 15, we gave nothing as a control. 15 scaffolds by themselves as a control. And 15 was the experiment, cells in the scaffold. Let me show you videos 100 days out. Here's a typical control, not our best, not our worst. And what I want you to focus on is their hind legs. And the two things to notice are that this animal is not able to bear his own weight. And notice the paws are splayed in a rather awkward fashion. So he's dragging them. You can actually score these. There's like a BBB score. It's about 5. And this is pretty typical, the mean of the control groups of about the 45 that we did, 100 days after the experiment started. Now let me go to the mean of the experimental group. This is the next to last slide. And what you can see with this guy, first of all, notice that he's kind of heavy, he's a bit fat, but he's able to bear his own weight even though he's eaten a lot. And by the way, this is no cure yet by any means. He's still clumsy and stuff, but he's big improvement compared to the controls. And you get the idea. Look at the paws, they're splayed in a much more normal fashion. It's a BBB score of about 13. Actually, my wife gives me advice. She says, don't leave that on too long. She gives me other pieces of advice, too. But one time I had that slide on for about 10 minutes, and she said, that's not good. Everybody's looking at the animal's butt. So anyhow, let me just summarize some of what I've talked about tonight then, about the potential. And really, I think potential is the right word. Nanotechnology is still at an early stage. But I think the potential is great. The potential could be a new drug delivery systems, to target drugs, existing drugs, to sites like a tumor. It could be a new drug therapies. The examples like I gave you with delivering DNA or siRNA, it may really enable them. You could create new materials, new tissues, and even new diagnostics and imaging agents. So I hope that it's an area that has a lot of promise. It's certainly been exciting for us to work on. I know there's some wonderful work out at UCSD on it as well, and all through the San Diego area. Anyhow, it's been an honor and a pleasure to speak to you tonight, and thank you so much for having me and for having this lectureship. My pleasure. My pleasure. Happy to.
We realize we're between you and dessert, but we do have time for a little questions.
Sure.
Roger.
For the siRNA delivery, I noticed you picked liver, which in some ways might seem to be the easiest target for the coital injury of some considerable diameter. Was there any other targeting agent put into the liver? And if not, how easily do you think this would be convertible to other tissues that are not so natural?
Right. Well, it's a great question. So what we did is you're absolutely right. It's a hard enough problem, so we tried to make it as easy as possible, and that's why we picked liver. One of the things, though, we have found already because we have this library, and you can use different ones for different tissues, is we have been able to get some lung targeting, and we actually are looking at targeting to other tissues. That's a big effort of what we're looking at. And some of the lipids seem to work better in non-liver sites, but we're still pretty early on that.
But there wasn't any deliberate, say, macromolecule put on the outside specifically to push it toward liver.
Correct.
No antibodies afterwards anyway.
No, you're absolutely right. It was just that one lipid that I showed you, and that's really been the strategy that we've been trying to use. Is can we, through this library, find different lipids that might work for other sites as well? But that's an excellent question. That really outlines a general direction we're going in.
In the same set of experiments with the combinatorial lipids, I call them combinatorial lipids, but the high throughput lipids that you did.
Either is fine.
You said that there was safety there, but do you know the metabolic fate of those lipids in circulation or in particular organs?
Yeah. So the question is, do we know the metabolic fate and the safety in the particular organs? I think the people at Alnylam do, but I probably wouldn't know it well enough to say. But I mean, they've done quite a bit of work on it, with respect to some of those issues. But I probably wouldn't be able to, say exactly what it is. Other than that, I think that they've found it's pretty good. But I just don't know all the details. Yes, there's one here.
One of the obvious portals for drug delivery is the lung, and one of the holy grails is obviously getting insulin without having to do an injection. Now, recently, there's been all these spectacular failures with Exubera being pulled by Pfizer, and I guess Lilly is pulled away from the air technology. I'm sure you're thinking about what happened, and I'm wondering, is nanotechnology a solution to perhaps the issues that arose with that?
Yeah. So the question that Rob's referring to is, there was all this work for many years on pulmonary delivery of insulin, and now we see it suddenly stop. What does that mean? Well, I think it's hard to know. I mean, you could talk to different people. I mean, my sense, and I think the answer is I don't think nanotechnology, I don't know what the answer will be. I think the problem is, depending who you talk to, a lot of people felt initially that, a couple things. When Pfizer was the big driving, I think a lot of these things are business and not science. So I think what happened is that Pfizer, and again, I'm only telling you different things that people told me, but I think Pfizer, felt that this was an opportunity, starting in the early 90s to get an insulin franchise that Lilly and Novo Nordisk had. And they worked with Inhaler, the Nectar, to do it. But that particular system ended up being a huge, bulky system, and I think that the marketing wasn't, according to at least the people at Nectar, particularly good. And so in the end, Pfizer, now we're in 2008 and companies want to have, multi-billion-dollar drugs to follow Lipitor, and the sales initially were not good. So they pulled out, and I think once they pulled out, it's hard for me to see that Lilly or Novo Nordisk, they weren't threatened anymore, why they wanted to, spend that kind of money. There were other reports about, possibly slight increases in cancer. I don't know. The problem is the statistics were so poor, it's hard to know. I mean, if they were the other way, you could say, well, it was curing cancer. But I think it's too early to, I think it's too early to know. So my feeling is when I look at that, it was probably more business driven than science driven. And in that sense, I don't think nanotechnology is an answer. I think the problem, sadly, with insulin, I mean, there's been all kinds of, good work that different people have done over the years with insulin. And, but the issue is any kind of small safety issue or change, when people feel it's not necessarily life-threatening, the FDA and so forth, and it ends up that, not being pursued, whereas, say, when you go after cancer, it does get pursued. I guess the last thing I was going to just say on the Nectar thing is that, I think that even though it's not maybe going to be useful for insulin. It may be useful for plenty of other things, and I think that aerosols could be useful for other things. But I'm not sure that you necessarily need to be nano to help on that. I think it's, a lot of other issues, to the extent that I know.
Thank you so much.
My pleasure. Thank you. Thanks so much, Adam. Yeah, well.